Hello, my name is Matt Veldman. I'm an assistant professor at the Medical College of Wisconsin in the Department of Cell Biology, Neurobiology, and Anatomy. My lab studies mechanisms of neurodegeneration and regeneration in the visual system. Specifically, we seek to understand the basic mechanisms underlying the disease glaucoma and identify ways to prevent and reverse blindness caused by this condition. Currently, at least 2 million Americans have vision loss due to glaucoma, and the incidence of this disease is increasing as our population ages. Blindness in glaucoma is characterized by age-related loss of peripheral vision and blurry vision in both eyes, progressing to complete blindness if not treated. Current treatments can slow the progression of the disease and preserve remaining vision. However, there is no cure, and lost vision cannot be recovered. Vision loss in the most common form of glaucoma is caused by damage to the optic nerve due to elevated pressure in the eye. The cells that signal from the eye to the brain, the retinal ganglion cells, are disconnected from the brain, resulting in loss of vision. These cells never regenerate their connections to the brain and often die. This is also true of all mammalian models of glaucoma. However, the small aquarium fish, zebrafish, has the incredible ability to regenerate the optic nerve and recover vision after nerve injury. My lab is using this model to understand the mechanisms of cell survival and regeneration after nerve injury to identify genes and signaling pathways that can be therapeutically targeted to improve vision in glaucoma. Zebrafish are amazingly 70% genetically similar to humans, and all of the known glaucoma disease genes are found in fish. The visual system of zebrafish and human is also conserved. The same cell types are present, and the tissue organization is similar. To identify genes involved in optic nerve regeneration, my lab has performed a genome-wide screen for gene expression changes during regeneration. We use 3-day post-injury as a time when robust regeneration is occurring, as you can see in this image of whole optic nerves circled in white with control on the left and regenerating on the right. A dotted line denotes the site of injury. The regenerating cell projections are labeled in green and can be robustly observed on the right image, but are absent on the left. We isolated the eye and optic nerve and used a technique called laser capture microdissection to isolate the different cell layers in the retina and assay changes in gene expression across the whole genome. We found that there were several thousand genes that both increased and decreased in the retinal ganglion cell layer and in the optic nerve, but there were relatively few changes in the other layers of the retina. Using bioinformatic tools and drug screens, we have identified several signaling pathways in both the retina and optic nerve that are active during regeneration. We have also identified some interesting fish-specific genes. Remember that 30% of the genes are not conserved between human and fish. These genes might mediate the differential response between fish and mammals to nerve injury. My lab is currently testing the importance of these genes and pathways for successful regeneration in the zebrafish and using mammalian models of glaucoma to determine if we can improve cell survival and stimulate regeneration. My hope is that discoveries made in my laboratory will translate into new treatment possibilities for glaucoma patients and make recovery of lost vision a reality.